So thank you, Yanni. By now you should see my presentation and also hear me. So shalom and hello everyone. It's a great pleasure for me to present to you some of my recent work, which is about the folding of DNA G quadruplexes observed by time-resolved NMR. So, and typically, if you would talk about folding of biomolecules, you would always show some kind of representation of your folding energy landscape. And the way I imagine folding energy landscape for nucleic acids, it's pretty much represented by this skiing region map. And the skiers of you will know that in the skiing region, you will have different slopes. And typically, you would have a blue slope, which is slow and rather family-oriented, easygoing. You will have the red ones, which are for the more advanced and also quicker and faster. And in the end, you also have black slopes, which are typically steep and narrow and are typically the fastest way down to the valley, down the hill. And of course, those different slopes represent folding pathways along the folding energy landscape. And what I'm going to show you today is an example of a single DNA sequence that is able to adopt quite similar secondary structures, but can occupy and go down 3 different of those distinct folding pathways. So to show you the basic architecture of G quadruplexes, it's quite easy to explain. So you have G nucleobases, which can arrange via Hookean base pairing to form this G tetrads. This G tetrads then can stack onto each other, and you end up with a basic scaffold of a G quadruplex. But typically those are intramolecular, and this means you just have a single strand, and thus results in different topologies, different polymorphic structures, which can be typically characterized by their relative strand orientation. And I'm going to show you with a pointer, the relative strand orientation, and also the resulting interconnecting loops. But in addition to this polymorphism, there's also non-conventional types of quadruplex polymorphism. And one of these features is G-register strand isomerism, which has recently been outlined by Tony Mittermeier and his group. And this arises if you have a G-rich strand, and there you can shift relative to the quadruplex core, one of the G strands, and you end up in different structures which can be characterized as G-register shifted conformation. We have recently examined such systems and also described the refolding dynamics of such systems. But what I'm going to talk about today is a different feature. And this arises in sequences where you have more than 4 nearby G strands, meaning that you have additional G strands that can eventually displace or exchange with one of the strands that is incorporated into the quadruplex structure. And such kind of folding isomers you can easily imagine as having a spare tire, and so the related spare tire dynamics come from this name. You find such sequences throughout the entire human genome, but a prime example for this is really the quadruplex forming sequence in the c-Myc oncogene promoter. I show you here the sequence representation in this kind of cartoon-like representation, and there's 3 different ways to assemble 4 out of 5 of these G strands into very similar, all parallel, but different stable G quadruplex structures shown here in this cartoon representation. And I show you here the 1D proton spectrum showing the fingerprint region of the quadruplexes, which is, you see here, the iminoproton resonances only of the iminoprotons that are incorporated into Hookean base pairing. And thus, the iminoprotons here are a very conformation-specific probe of your conformation. And what you see in this wild-type sequence is that you have a coexistence of all 3 different conformations. Yes, you have a major conformation, but the others are also present. And the essential idea how we tried to disentangle and separate the underlying folding mechanisms for all 3 different conformations is by introducing photolabile, meaning light-sensitive protecting groups. And thereby we can block specifically the Hookean base pair interaction site and thus prepare transiently non-equilibrium states of a different nature. And upon wavelength selective irradiation, you can selectively then photolyze the protecting groups and thereby fully restore the native state of your DNA. And this is set up how we do this in Frankfurt, which is quite also easy to explain. It's no rocket science. You have a high intensity laser light source, which is coupled via glass fiber directly to your sample within the NMR spectrometer. And this allows for in situ laser irradiation. And this gives a trigger dead time down to the sub-second scale. And the way now to separate and isolate the three different conformations out of the polymorphic ensemble is by introducing permanent nucleobase replacements in one or the other strand, which will prevent the incorporation into the G-quadruplex, so you can select different conformations. And in a second step, then you can introduce multiple photo cages, and this will prevent the DNA from any kind of base pair interactions or overall folding interactions. And I show you here the basically empty or baseline fingerprint region of all three different conformations, meaning that at physiological sample conditions, you have a completely unfolded state. And after light irradiation, then you quantitatively restore the native fold of the respective conformations. And thus are the related conformation-specific kinetics of the folding reaction. And I show you the kinetics as a function of the build-up of the aminoproton signals. And then you can do, of course, bioexponential fitting, and this will yield you the respective rate constants. The interesting part comes now if you look at the folding in a temperature-dependent manner. And I show you here the Arrhenius plots of all three different conformations. And even from first sight, you can tell that they have inherently different underlying folding driving forces and underlying folding mechanisms. I'm going to explain to you that step by step. So the first case is quite easy to explain. Here you have the single conformation with a negative energy activation energy barrier for both rate constants. And this can be easily explained if you imagine that this conformation is going down the black slope. So you have really optimized kinetics. You just go down the folding pathway. You don't have measurable population of any on or off pathway intermediates. And this follows kinetics you would expect typically for protein folding, which is a funnel-like folding mechanism. But the opposite case is true for the second case. Here you have a positive activation energy barrier, meaning that you have during folding some kinetic traps which have activation energy barriers that you have to overcome in order to refold those traps. And if you examine the folding reaction now at lower temperatures, I show you here the buildup of the major conformation. This is the kinetics in green. But in addition to that, you can also detect signals which give rise to a transient intermediate kinetic trace shown here in red. And after careful analysis of our kinetic data, we came up with this kind of kinetic model, which perfectly explains our experimental data. This model could be further supported by an independent experimental approach where we measured the kinetics of folding with time-resolved optical spectroscopy, which is CD spectroscopy in this case. And this yields you the exact same kinetics for the intermediate formation, but in addition gives you some more structural insight about the nature of the intermediate state. And taken together our spectroscopic data, which are in line with observations made by mass spectrometry and also from predictions from molecular dynamics simulations, we can come up with a structural prediction, a structural model, which is pretty much represented by this half-baked G-quadruplex conformation. But however, this intermediate state is an anti-parallel topology, meaning this has to be completely unfolded and refolded again in order to transform into the major conformation. And this gives rise then to the positive activation energy we see in the experiments. So the underlying folding mechanism is here real kinetic partitioning, and we see off-pathway trapping with intermediate states of lifetimes of up to several minutes. But certainly the most peculiar case is the third one. And what you see here is a strictly non-Arrhenius behavior. And this is really a rare case where you are in the intersection on the folding energy landscape, meaning that you have two different folding regimes at higher and at lower temperatures. And to explain this non-Arrhenius behavior, I show you again the prediction we made, the structural model for the intermediate state. So in this blue conformation here, this intermediate is stabilized by the resulting internal two nucleotide loop. And this follows also from MD predictions that this is optimal now to stabilize this conformation. Following this, also this explains why we are here on the very fast funnel-like folding, because here the resulting single nucleotide loop prevents a stable formation of this intermediate state. Thereby you bypass this intermediate state and directly fold down to the major conformation. And following this argumentation, the intermediate state for this long looped, this red conformation here, this would result in a long internal 6 nucleotide long loop. And this yields or results in kind of a frustrated double hairpin intermediate state, which at higher temperatures is easy to overcome and find into its global minimum. But at lower temperature, this gets really a real kinetic trap. And this explains the bipartite kinetic behavior of this conformation. So in summary, what I've shown you, and this is a different representation of the skiing region map, is I showed you the 3 key steps of the 3 major folding pathways leading to this polymorphic G-quadruplex ensemble. And there are, of course, additional features, which is refolding and also different folding pathways, which I was not able to talk about today, but I'm happy to share in the breakout session with you. And in the end, I have to say, because I have a different affiliation now, this work has all been done during my PhD in the group of Harald Schwalbe in Frankfurt. And I would really like to take the chance to acknowledge and thank all the people who were involved into this project, and also, of course, acknowledge funding now for the postdoc funding to take up some of the projects here at Weizmann. And last but not least, I really want to thank the organizers for this great webinar series and also for the opportunity to speak here today. Thank you.
Thank you very much for the really exciting talk.
Okay. Good morning, good afternoon, good evening, good night to everyone. First of all, I would like to thank Angelo and other organizing committee members for really putting up this great platform for us to share with you some of our exciting results on the room temperature over Hauser DNP enhanced carbon and mass spectroscopy of small biological molecules in water. I'm Jiafei Mao from University of Frankfurt in Germany. So in the recent years, we've all been seeing how DNP, dynamic nuclear polarization, as a powerful technique has transformed the solid state and mass spectroscopy. Just by picking up a number of examples here from my own research, it's clear that this technique is able to help solid state and mass spectroscopists to answer a wide range of highly challenging biological questions on very difficult targets, for example, membrane proteins and ribosome nascent chain complexes. In solid state DNP, often we are actually rather spoiled by the low temperature, about 100 Kelvin, introduced by the cryogenic magic angle spinning condition, which on one side resolves a lot of technical difficulties. On the other hand, actually compromises the spectral resolution, especially for the highly dynamic biological molecules. So it would be great to migrate back to the room temperature scenario, and not to mention that at room temperature, there's a huge solution NMR community also waiting for DNP to boost the sensitivity of their spectrum. However, at room temperature, we have to face many additional issues from microwave sample heating, microwave efficiency problem, which is related also to the small sample size. All these point to a highly challenging engineering problem that is microwave RF resonator. On the other hand, because the molecules are highly mobile at room temperature, so we have to use a very restricted mechanism, in this case, the Overhauser effect, to transfer hyperpolarization from the electron spin to the nuclear spin, which rely on actually the ultra-fast intermolecular dynamics that is really underexplored topic in general in chemistry and condensed matter physics. So it behaves like a black box to the community. So here in Frankfurt, my colleague Vasio in Thomas' group over years has built a beautiful instrumentation platform at 9.4 Tesla, which allows us to perform the DNP experiments in liquid at this relatively high field. However, at the moment, this instrumentation platform is really technically complex. Namely, it's a bit difficult or even tricky to perform the experiments there. So we find ourselves often trapped in a double dilemma. On one side, it takes a huge amount of time and efforts to run the experiments. On the other hand, actually, we have no idea basically which molecules will be the proper target for this technique. So it would be great, on the other hand, to understand a better way to fish out or to search the proper molecular targets, to have some guidelines. So several years ago, Guoquan and Marina's group in Göttingen has made a breakthrough scalar Overhauser DNP carbon NMR spectroscopy. They have found that the carbons in a number of simple small organic molecules can be hyperpolarized directly in organic solvents at relatively low field, 3.4 Tesla. But how can we migrate this kind of approach to a more challenging platform? So for this, we first need to go to the very bottom of how electron spins crosstalk with nuclear spins. Of course, we understand their interactions involve the dipole interactions and the scalar interactions. And here in this talk, we will restrict to the scalar interactions, namely the electron spin density dispersed to the position of a particular nucleus position. This is so-called Fermi contact. This kind of a delocalization depends on the molecular orbital. So obviously we need a chemical process. There is a chemical interaction between the polarization agent, for example, the radical, and your target or substrate molecule. And when this interaction fluctuates at a proper frequency, we'll take a look at this in more detail in a few minutes. This could drive the zero quantum electron spin, nuclear spin cross-relaxation process. So here we have the famous 4 energy level diagram, and we shine the microwave to the electron spin to repopulate the electron spin at EPR energy levels together with the zero quantum cross-relaxation pathway, you obtain DNP enhancement, for example, in this case on carbon. And to understand this process more quantitatively, we of course have to go to the famous Solomon equation. Due to the time limit, I'm not going to dive to the details. Nevertheless, by this analysis, we understand that this red term called a coupling factor determines Overhauser DNP enhancement. And within this red term, of course, the cross-relaxation rate is the decisive factor. So from the previous slides, you already may understand that the cross-relaxation rate, of course, is determined by the hyperfine interaction between the electron spin and nuclear spin. And here we dissected these two aspects, the scale or magnitude and the dynamics of the hyperfine interaction are very important to determine the cross-relaxation rate and determine the performance of Overhauser DNP. So let's take a look at the magnitude of the hyperfine interaction first. Indeed, a few years ago, Haridon and co-workers has demonstrated beautifully that the DNP enhancement can be correlated to the hyperfine coupling constant. And we realized that the Fermi contact actually has 3 distinct but highly related spectroscopic consequences. In DNP, of course, it drives the scalar Overhauser DNP enhancement. In EPR, this generates the famous hyperfine shift. And in my beloved paramagnetic NMR field, actually, this leads to the famous contact shift. And the connection between DNP and EPR, EPI, and paramagnetic NMR has been extensively explored in literature, but there are not too much reports connecting the paramagnetic NMR shift and the DNP enhancement. So I wonder if actually the paramagnetic NMR shift can be used as an indicator to predict the DNP enhancement and to have a guideline. And taking this hypothesis, I dived into quite older literatures, mainly published from in 1960s to 80s, paramagnetic NMR on the radical small molecule interactions. And from this particular publication on JAX in 1984, actually the authors has demonstrated that by paramagnetic carbon-13 shifts, that this particular molecule called imidazole interact with temporal radical. So we put this molecule in the D2O and repurposed this paramagnetic NMR study to the high field DNP. So we used our home-built Fabry-Perot resonator for people who are more interested in the instrumentation details and some related applications. You may also follow some coming presentations from Thomas Prisner's group and my own talks on the Euromap. Nevertheless, as shown on the left, we obtained about 50-fold DNP enhancement on the carbon on this molecule in D2O at room temperature, which is truly impressive. This encouraged us to explore some other biological molecules, for example, glucose in water. And again, we do paramagnetic titration experiment by varying the radical concentration, and you can see the presence of paramagnetic carbon chemical shift by this titration experiment, which informs the DNP experiment at a high field. And pleasingly, we indeed observed very nice DNP enhancement on most of the carbons here. And again, we further encouraged to explore other water-soluble small biological molecules, for example, water-soluble amino acids. And for serine, for example, we can also do this titration, and the paramagnetic NMR shift informs the DNP performance again. And as you can see here, the aliphatic carbons are quite enhanced. So this can be expanded to other water-soluble small molecules like alanine and glycine. And same approach, first we run the paramagnetic NMR experiments as a pre-screening and then subject to the technically more complex high-field DNP measurements. And we consistently see the nice enhancement on many carbon sites on these molecules. And even on the structurally more complicated proline, and we do see all the aliphatic carbons are quite enhanced. So here you see several interesting features. First is actually our room temperature solution DNP NMR experiments shows quite a good resolution. Even the J coupling can be well resolved. So this really outperforms the resolution of the conventional low temperature DNP solid state NMR spectroscopy as shown here. So we really start to deliver the promise that the resolution would be improved at room temperature scheme. On the other hand, you do see that the carboxylated group carbon shows very low DNP enhancement compared to the aliphatic carbons. And we do observe this kind of phenomenon on all the amino acid we have tested. To understand this type of site specificity or selectivity of the DNP enhancement, I called my friend Xiaohe in Shanghai, and they performed the DFT calculations on number of the amino acid temporal complexes, and our simulation clearly indicates that the temporal radical actually can interact with many side chain carbon sites of different amino acids through the CH-mediated intermolecular hydrogen bond, which explains, of course, the observed DNP enhancement on all these side chain carbons, but also the paramagnetic NMR chemical shifts. So up to now, we've been only talked about the scale or the magnitude of this interaction, so namely how strong it is. But of course, to drive efficiently the electron carbon zero quantum cross relaxation, we need to fulfill certain time scale or frequency regime. And by taking 9.4 Tesla as our experimental parameter, we can derive that the proper dynamics to drive efficient zero quantum transition should be at sub-picosecond time scale. So we would like to understand what is the correct molecular dynamic mode or features really drives the Oberhauser DNP enhancement we observed on the sample. So for this, we actually took a very different approach from the literature. We call it the bottom-up or forward approach. Namely, we do the extensive QMM simulations of our molecular target. So we can get an explicit picture of the dynamics and extract the dynamic features that are relevant to the Oberhauser DNP performance observed on the macroscopic samples. So for this, we need obviously a chemically more defined system. So we went back to the original paramagnetic NMR paper published on JAX in 1984 and figured out this particular molecule called indole also interact with the temporal type radicals. And indeed, we can see by DNP experiments that there are quite good DNP enhancement on most of the carbons and even the site-specific DNP enhancement again on this molecule. The nice thing of this molecule is the chemical interaction between indole and temporal radical is rather defined through single binding site. So this encouraged Xiaohe's team really to perform heroic QMM simulations of relatively long trajectory at high level. So what we can confirm first is there is, of course, the NH bond mediated intermolecular hydrogen bonding between the temporal radical and the substrate molecule indole. But we also observe significant dynamics of this complex, namely the intermolecular hydrogen bond fluctuate, but also the ring structure fluctuate all the time. So obviously, this kind of structural rearrangement would tune the anti-pi orbital overlapping between the temporal radical and the substrate molecule, where the electron spin density actually sits on. So this in turn would tune the spin density on different indole carbons. Now, by analyzing the spin density of these carbons, we can calculate or covariance matrix or the distribution. We find actually kind of the individuality of all these fluctuations on different carbons. So this encouraged me to dive into more comprehensive models by taking the spin density trajectory from our QMM simulations and run through the analysis by calculating the autocorrelation function, spectral density function, but even more to calculate the memory function that is not well used actually in magnetic resonance field. So to cut the long story short, that our simulations clearly indicate the presence of a number of terahertz phonomodes in this hydrogen bonded complex, which could contribute to the Oberhauser DNP behavior we have observed. Now, by analyzing spin density trajectory using the autoregression model that is very well loved by the statisticians, or we take the autocorrelation function and analyze this trajectory, this function using the non-Markovian memory kernel hypothesis, we can all confirm the presence of a femtosecond time scale short membrane in this dynamics. And very nicely from the autoregression model, we also understand that this very, very short membrane, which is possibly dictated by the quantum mechanical instructions, actually would cast the slower phonomode, terahertz phonomodes, which contribute to the Oberhauser DNP performance. So now actively we are analyzing the nature of this short membrane kernel. But the experimentally observed DNP enhancement actually comes from many other contributions as well. We talked about mainly the scale of Oberhauser DNP, but we also have the experimental evidence to show at the same time there are different dipolar Oberhauser DNP mechanisms. And in this particular chemically engaged complex, the classical point dipole approximation can even not be held. So we really start to see a highly complex picture, and not to mention the negatively enhanced proton and positively enhanced carbon could even still crosstalk with each other through the intramolecular heteronuclear NOE process. And at the moment, thanks to the dual channel setup we recently established in the lab, we're actively investigating also this pathway. So with this, I would like to draw you to my conclusion that it's clear that now the room temperature carbon Oberhauser DNP experiments can be performed at least on small biological molecules in water at high field. And I hope I've convinced you that paramagnetic NMR spectroscopy and QM level simulations can facilitate liquid state or DNP discovery. So they are indeed good friends of us. And it is the time to embrace the chemical diversity and the mechanistic complexity of Hauff-Haus DNP in liquids. So with this, I would like to thank all my collaborators, especially Thomas, Stanhua, and Vasil for their contributions on the experimental side, and the Xiaohe's team for really a lot of efforts on the computational chemistry side. And I thank you for your attention. Cheers.